mitosis and meiosis, there is one kind of question in which many of you feel difficulty, and that is calculation of number of chromosome, chromatid, centromere, DNA molecule, etc. So to answer such kind of question, there is a very simple table which you can go for mitosis, meiosis both, and through which you can answer all kind of questions. So let's start with mitosis. So in mitosis, if we say, so in mitosis we will first see all the stages, right, and then accordingly we will see the different number of chromosomes. So mitosis, G1 phase, S phase, G2 phase, then we have prophase, we have metaphase, we have anaphase, we have telophase, and cytokinesis, right? Then this side, let's see, question can be regarding the number of chromosomes, or the question can be regarding the DNA content, right? So let's see these two first, right, and rest you can easily find out with this. So let's make it in a form of a table. So let's see here, line easy. So now if I have to see it, so first of all, if I ask you what is the number, just a second, first complete this. Now let's see, mitosis mostly take place in 2n, right? The cell in which mitosis take place, these cells are 2n. So in G1 phase, we will start with 2n, right? Although mitosis can take place in haploid cell also, but mostly it is diploid. So if it is 2n and DNA content here will be 2c. Now how a chromosome will look like here? So chromosome will something look like this. Although very clear, I want to say that in G1 phase there is no visibility of chromosome. Chromosome doesn't appear like this. Just for your understanding, I am telling that if chromosome existed, it will appear something like this. Then in S phase, DNA replication will take place. Now when the replication take place, see, if this is chromatid, so after replication it will become like this. So what you understand here, that here you can see there is one chromatid and here the chromatid is 2, means replication has happened, right? But still here there is one chromosome, this is also one chromosome. So number of chromosome remain the same, but DNA content become double. G2 remain the same, 2n, 4c. Prophase remain the same, 2n, 4c. Metaphase remain the same, 2n and 4c. Now in anaphase what happens? So till metaphase things are like this only, right? One chromosome with 2 chromatid. But in anaphase what happens? Centromere split. It is split like this. It splits and both the sister chromatid move toward the opposite pole. Now when it is split, see here one chromosome if I count is, but this will be 1, 2, right? So number of chromosome is getting doubled in anaphase. So in anaphase the number of chromosome will become 4n and DNA content remain 4c. Finally, telophase and cytokinesis will take together because they happen side by side, right? So by the end of cytokinesis we get 2n and we get 2c, right? So 2n and 2c. So what you see, it starts with 2n and 2c and it ends with 2n and 2c. Now if you are wondering what will be for telophase, see, if we take telophase separately, then in telophase also it remains 4n and 4c. But usually telophase and cytokinesis take place together, so that's why it appear ultimately like this. So now you understand this part. Now anywhere if you are given chromosome of any stage, you can, let's say if I give you the number of chromosome of anaphase, and if I ask you about G1, so half of anaphase. If I give you G1, ask you metaphase, then you will say equal. So let's see one example. If I give you number of chromosome in G1, so G1 has 18 chromosome. Now tell me the number of chromosome in metaphase. So G1 has 2n, metaphase also have 2n. So I will say metaphase also will have 18 chromosome. But if I ask you anaphase, so it will have double the chromosome, 36, right? At the end of mitosis, so I will say 18. Same way if I ask you DNA number of chromatid. So if I ask you number of chromatid, so we know that, see here you can see one chromosome has only one chromatid, but here one chromosome has 2 chromatids. So here number of chromosome in G1 phase, number of chromosome and chromatid will be the same. But again, if it is not visible here, so usually that way question is not asked. So it will be asked like number of chromatid in anaphase. So anaphase will be again 36 chromatid. So here chromosome also 36 and chromatid also 36. But in metaphase chromosome is 18, but chromatid is 36. See, chromatid is 4c, right? So everywhere here, if I say wherever is 4c, the chromatid will be 36. Wherever it is 2c, chromatid will be half of it. So this point clear. Another important point, if I ask you, sometime question is asked about number of centromere. If question is asked about number of centromere, so remember, number of centromere is always equal to the, so number of centromere is always equal to the number of chromosome, right? So very simply you can now easily do that also. Then if sometime another type of question is asked, that is the DNA content. Question can be asked, tell me the number of DNA molecule. Number of DNA molecule. So number of DNA molecule is equals to number of chromatid. As much number of chromatid, that much will be the DNA molecule. So for example, if I ask you chromosome means a cell has 18 chromosome. A cell has 18 chromosome in G1 phase. Tell me the number of DNA molecule in. Tell me that number of DNA molecule in NA phase. What will be the DNA molecule in NA phase? So simply, now you have to find the number of chromatid in NA phase, right? So the number of chromatid in NA phase is 36. So DNA molecule will also be 36. Very easy. So same way, number of chromosome, number of chromatid also you can easily calculate. Then sometime DNA content is also asked on the basis of picogram. Sometime they will give you, let's say, 8 picogram in G1 phase. So how much will be in NA phase? So NA phase will be double of that. So 16 picogram. Right? What if 8 picogram in G1 phase at the end of mitosis? So you'll say it is the same. So at the end of mitosis also 8 picogram. So in this way, simple question can be asked. Remember, question can be asked on the basis of chromosome. You can easily do by counting the 2n and n or 4n. Then question can be asked about number of chromatids. That also you can do easily. Then question can be asked about number of DNA molecule. That also now you can easily do. Then question can be asked about number of centromere. That also now you can easily do. So these are some of the basic questions which will be asked from mitosis. Now let's move to the meiosis. Now we know that meiosis, there is one replication and two division, right? So let's say here G1 phase, S phase, G2 phase, then prophase 1, then metaphase 1, anaphase 1, telophase 1, and telophase 1, that followed by dyad of cell, then prophase 2, metaphase 2, anaphase 2, and finally telophase 2. This represents the tetrad of cell, right? So this is the overall meiosis, in which we say that this much part is meiosis 1 and this part is the meiosis 2, and these represent the two division, and here is the replication, right? Replication happened during interphase, specifically during the S phase. Now, but I am not going into detail of that you all must have already studied properly. So again, here I will focus on the number of chromosome and DNA content. So let's see here. Now, if I ask you number of chromosome in G1 phase, so what you will say? So 2n, right? DNA content here 2c. Then in S phase, replication happened, but the number of chromosome remain the same, 2n, and here it will become 4c because replication happened. 2n here become 4c, right? Remember, this is DNA content and this one is the number of chromosome. Now in prophase, continue 2n, 4c, 2n, 4c. Now in anaphase, see, in mitosis, in anaphase the chromosome number become double. Why? Because there is splitting of centromere happen. Is there any splitting of centromere in anaphase 1? No splitting, so the number of chromosome remain the same, 2n, 4c. Now here telophase, it form dyad, so in the dyad it will be n and 2c, right? Then it is followed by a very short phase which is called interkinesis, right? And then prophase 2, so this dyad will enter into meiosis 2, so it will remain n and 2c. Then metaphase n and 2c. Now what happened during anaphase 2? Splitting of centromere will happen, and splitting will increase the chromosome number, so it get double, 2n and 2c. And then in tetrad and telophase, 2n become n and c. So as you can see, 2n, 2c in the end become n and c. So can we call it reductional division? So this will be called as reductional division. So again here the same way question can be asked from number of chromosome, number of chromatid, you can easily calculate from here. For example, if I ask you this statement, see very carefully and tell me, number of chromosome in anaphase 1 is equal to number of chromosome in anaphase 2. So it is correct. Number of chromosome in dyad of cell is equal to number of chromosome in tetrad of cell. Again true. If I ask you the DNA content of anaphase 2 is half the DNA content of anaphase 1, correct? If I say the DNA content of diad is double the DNA content of tetrad, that is also correct. So in this way questions can be asked. Now another statement, if I say the number of chromosome during metaphase 1 is double as compared to number of chromosome in metaphase 2, yes, it is also correct. So now if I give you about G1 phase, you can easily calculate anywhere. So just quickly let's see one example and then we will move to the next part. So in G1 if I say there is 8 chromosomes, right? So if I ask you in anaphase 1 how many chromosomes? 8, right? So number of chromosome in anaphase 1 remain the same, 8. In anaphase 2 if I ask you 8, but if I ask you in tetrad, so tetrad become half, so 4. In dyad it become half, so we say 4, right? In metaphase 1, so it remain 8, but in metaphase 2 it become 4. Is it clear? Now, so this point is very much clear. Then number of chromatid, again the same thing you can see, right? One more question which is asked, that tell me in which stage of meiosis, or in how many stages of meiosis each chromosome has only one chromatid. See here, if we see the entire meiosis 1, each chromosome has how many chromatid? It has 2 chromatids, right? Even in prophase and metaphase each chromosome has 2 chromatid, but in anaphase 2 splitting of centromere happen. So here, so if question is asked that dyad have, in dyad each chromosome has 2 chromatid, but in tetrad each chromosome has only 1 chromatid, so it is correct, right? Now one question here I am giving you. If I say that a gamete has 18 chromosome, a gamete have 18 chromosome. Now tell me the number of chromosome at each pole of anaphase 1, at each pole of anaphase 1. See, when I say gamete has 18 chromosomes, so how many, what is the DNA content of gamete? So gamete is n, means I am giving you n is equals to 18, right? Now from this table you can easily find out A1 means anaphase 1 is how much? 2n, right? So number of chromosome in anaphase will be 36, but question is not asking number of chromosome, it is asking at each pole, right? So during anaphase, if this is the cell, at each pole we have n and n, right? So at each pole how many? 18. At each pole there is 18 chromosome. So in this way such questions can be asked from here. Same way here also DNA molecule number can be asked. So the relationship between the number of DNA molecule and the chromatid remain the same here. If question is asked from the centromere, it will also remain the same here. So both these table, right, as you can see here for meiosis and mitosis, if you remember this table, you can easily answer all kind of question related to this, right? So just quickly go and solve at least 10-15 questions based on this, where number of chromosome is asked, DNA content is asked, chromatid is asked, DNA molecule is asked, centromere is asked, all such question completed, right? So that is it for this topic. Quickly I will go through some more topics which will be very helpful for your NEET.